Hello everyone, I'm myself Dr. Dipti, Kariya, I'm PhD Physiology and your educator for Physiology. Today we'll discuss about functional anatomy of liver as well as biliary tract. Before starting my class, I would like to inform you all that you will get all my notes in soft copy as well as hard copy. To get soft copy, you have to download the application, the Serious, and subscribe for it. And for hard copy, you can order my book that is available on Flipkart and Amazon. Link for Flipkart, Amazon as well as my application, they all are there in the description box. You can also join a WhatsApp group. The number is 78638226668. We will try to solve your queries regarding physiology and other subjects of the first year. Also link for the WhatsApp group joining that is also given in the description box. Now let us start our today's topic, that is functional anatomy of liver. Okay, you all know that liver is the largest gland in the body. Weight is about 1.5 kg. Okay, and that is located under the diaphragm. You can see within the rib cage in the upper right quadrant of the abdomen. You can see here this is the location. This is the anatomy of liver. Now liver consists of 4 lobes, 2 major lobes, left lobe and right lobe. Okay, and minor lobes, they are caudate lobes and quadrate lobes. Okay, this one. Now right lobe, that is larger one, and left lobe is smaller, and this is smaller one. Okay, ducts, liver has got common hepatic duct, cystic duct, that is from gallbladder, they unite to form common bile duct. We will discuss in detail. Okay, now so right and left functional parts of the liver, that is right and left lobe. Okay, they have their own blood supply to the hepatic artery. You can see here. Okay, portal vein and their own venous and biliary drainage. Okay, liver has got physiological reserve, and also it has property of regeneration. It has regeneration power. It restores within 6 to 8 weeks of the removal. If you remove the liver, most of the liver if we are removing, then within 6 to 8 weeks it gets regenerated. 3/4 of it gets regenerated. So these are the peculiarities of liver. Now structural characteristic. Liver tissue consists of about 1 lakh hexagonal cells, hepatic lobules. This is one hepatic lobule. Okay, and this is made up of columns of hepatic cells. These are all the cells, hepatic cells. These hepatic cells are known as hepatocytes. And these hepatocytes, they are arranged in the form of a 1-layer thick plate. Okay, you can see here, these are the hepatocytes. You can see here. Okay, 1-layer thick plate is there. Fine. Now next is, in between these hepatocytes, you can see there are canaliculi. This canaliculi through which bile passes. So this is known as bile canaliculi. Okay, and hepatic cells, they tunneled by a communicating system of lacunae, and this is known as sinusoids. So you can see here, this is canaliculi here through which bile passes, and these are sinusoids through which blood passes. Then this sinusoids, they open in the central vein. You can see here. Okay, so direction of movement of blood is this side, and direction of movement of bile is in this direction. Fine. Now blood sinusoids, they are lined by endothelial cells. You can see here, these are sinusoids, and these are the endothelial cells. This all. Okay. Okay, and they also have macrophages here. Macrophages, they are our phagocytic cells. In liver, macrophages, they are known as Kupffer cells. These are very important. Macrophages means they have phagocytic action. If any foreign particle comes to liver, that gets phagocytosed by this Kupffer cells. Okay. Now this endothelial cells, another important characteristic here is endothelial cells, they are fenestrated. They have large fenestration, and they forming intimate contact between blood and hepatic cells. Because of this fenestra, these hepatic cells are in contact with the blood, and this helps the liver to transform or modify many of the constituent of blood. Okay, because of this fenestration. Okay, along the periphery of each lobule, you can see here, this lobule is hexagonal. You can see here, this is one, and at the periphery, at each corner, you can see hexagon, so it has 6 corners. So at the corner, we have portal triad. Triad means 3 things. It has got branch of portal vein. This is, you can see here, portal triad. This is portal vein. branch that is portal venule. Okay, branch of hepatic artery. Red one is branch of hepatic artery and bile duct. Okay, these three things they form a triad, portal triad. Okay, now blood from the branch of portal vein and hepatic artery, as I told you, they enter the sinusoid and it drains to the central vein. Here, this way, this is the direction of the blood from portal vein and hepatic artery. This one. And this is the central vein in the center of this your lobule. Okay, so you can see here, this is the direction. You can see here, this way towards the center. This is the direction. Fine. Now there is also another thing, that is space of Dis, space of Das or space of Des, that is between or beneath the endothelial cells. These are hepatocytes and these are endothelial cells. Okay, so between or beneath, between hepatocytes and endothelial cells, we have space of Des. Okay, and this is also known as perisinusoidal space. This sinusoids, this your blood vessels, they are porous and therefore they are known as sinusoids also. Okay, so this is perisinusoidal space, and it possesses little resistance to the movement of molecules. Even large molecule like albumin also, they can pass through this spaces. Okay, and this space connect with the lymphatic vessels too. Now, blood supply. Liver receives blood from mainly two sources. One, that is portal vein, and second one, that is hepatic artery. Okay, now you can see here, this is the portal vein. Okay, this portal vein, that is formed by union of two veins. Number one, that is your superior mesenteric vein. You can see here from mesenteric, here superior mesenteric vein, and second, that is splenic vein. Okay, the superior mesenteric vein drain blood from the stomach and intestine, and this blood, it contains digestive product like monosaccharides, amino acids, digestive products. They reach to the liver. Okay, and they also have bacteria sometimes along with this blood from the digestive tract. And splenic vein, it drains blood from pancreas and spleen, and this blood is rich in the insulin coming from the pancreas. Okay, then second one, that is hepatic artery. You can see here, this is the hepatic artery. Okay, hepatic artery is, it contains arterial blood. Okay, and hepatic artery only supplies 20 to 25%. Major supply, that is by portal vein. Okay, so this hepatic artery, it is a branch of celiac artery. You can see here, this is here. Fine, and it supplies 20 to 25% of total hepatic flow, 300 to 400 ml per minute, and it carries metabolic, it caters metabolic requirement of the liver tissue. Fine. Now, both these vessels, hepatic artery and portal vein, okay, they break into large number of branches. Okay, and these branches are given at each and every year to the portal triad. Okay, these branches of portal vein and hepatic artery, they get mixed in the sinusoids, and the blood runs the rows of the liver cells in the hepatic lobule. Okay, so liver has got metabolic products, or you can say products of the absorption from digestive tract, as well as oxygenated blood too. You can see here, these are the sinusoids. I will show you, these are the sinusoids. Okay, this one you can see here, this is bile duct, this is hepatic artery. Okay, and you have portal vein branches, and they pass through this sinusoid to the central vein. Okay, large molecule also can easily pass, as we have discussed. Okay, from sinusoid, because sinusoids, they have fenestration. Fine. So even protein molecules, plasma proteins also, they can easily pass from blood to the hepatocytes. Fine. Now sinusoidal blood that drains into the central vein, as I told you, this blood gets mixed and this will drain to the central vein. Okay. Now this central vein, from central vein, ultimately it forms right and left hepatic vein. You can see here, these are hepatic veins. Okay, and this hepatic veins, they open in the inferior vena cava, and that will go to the heart, then it will go to the lungs for oxygenation. So this is the circulation. Now, next is hepatic biliary system, that consists of two systems. Number one is intrahepatic, inside the liver, intrahepatic biliary system, and second is extrahepatic biliary system. Now, so starting with first, intrahepatic, inside the liver biliary system, as we have discussed, bile is secreted by hepatocytes. Here, these are the hepatocytes. Okay, they drain into the bile canaliculi, this one. Okay, and this bile canaliculi, they you can see through that it reaches to the tributary of the bile duct here, which is present in the portal triad. Okay, we have discussed portal triad is located here at the each and every end of this here. In portal triad we have branch of hepatic artery, branch of portal vein, and tributary of bile duct. Okay, now after that you can see here from bile canaliculi, whatever bile that passes and that enters into the bile canaliculi, then all they unite, these canalicules, they unite to form large duct, and ultimately they join and they form right and left hepatic ducts. Okay, so up to this we have intrahepatic circulation. Then they will next circulation, that is extrahepatic, outside the liver. So up to hepatic ducts we have inside the liver, this bile, it is there. Okay, then extrahepatic circulation, that consists of gallbladder and other bile ducts, we will discuss. Okay, so gallbladder, you can see here, this is gallbladder, that is thin walled pear shaped sac that is located at the inferior surface of the liver. Its function is it stores the bile when it is not required. Okay, when the digestion is not going on, we don't require bile. Okay, and it causes controlled release. Along with that, it also causes absorption of water and electrolytes and concentrate the bile. Capacity of storage of gallbladder, that is about 30 to 50 ml, and it has got three parts: fundus, body, and nape. We will discuss in detail later on. Now, here we will discuss about extrahepatic ducts. You can see here, right and left hepatic ducts are there. This right and left hepatic ducts, they unite to form this one, common hepatic duct. This common hepatic duct is about 4 centimeter long. Okay, then again you can see this is the easy diagram, you can easily remember right and left hepatic duct, common hepatic duct. Okay, 4 centimeter. Then to this common hepatic duct we have a cystic duct that joins this one, and this joining of cystic duct that originates from the neck or from the gallbladder, and this is also 4 centimeter long, this one, cystic duct, and this joins with your common hepatic duct, and this joining of common hepatic duct and cystic duct, it forms this common bile duct. Okay, here this common bile duct now we'll discuss. You can see here these are right and left hepatic ducts. They join with the cystic, they join to form common hepatic duct, then common hepatic duct joins with the cystic duct and they form common bile duct. Now this common bile duct, that is about 8 centimeter long, it joins with the pancreatic duct. This is pancreatic duct. Both of this they open in the second part of duodenum. Here there is dilated duct. These two unite and they form a dilated part that is known as hepatopancreatic ampulla or hepatopancreatic duct. Okay, this is also known as ampulla of water. Okay, we have discussed with the pancreas. Okay, this ampulla of water that opens the second part of the duodenum. Okay, now terminal part of the ampulla of water has one sphincter, and this sphincter is known as sphincter of Oddi, not properly seen here. Okay, and sphincter of Oddi plays an important role in controlled release of bile as well as pancreatic secretion. You can see here there is a sphincter. You can see main pancreatic duct and sphincter here. So this sphincter of Oddi regulates the secretion of bile and pancreatic duct secretion. Okay, so this is all about extrahepatic and intrahepatic circulation. Fine. So this is all about our today's topic, functional anatomy of, you can see here, this is the sphincter of Oddi. Can be seen properly here. Fine. So this is all about our today's class. You can get all my notes in soft copy and hard copy. As I repeat, for soft copy you have to download my application Vesalius and subscribe for the same, and for hard copy you have to order my book that is available on Flipkart and Amazon. Name of the book is Physiology Notes. Okay, link for all that is given in the description box. You can join our WhatsApp group. The number is 78638226668. Also you can join our WhatsApp group by the link that is given in the description box. Thank you so much. If you like my video, you can like it, you can share it with all your friends and subscribe our channel. Thank you so much.